The Multi-modal Imaging Genomics Transformer (MIGTrans) is a novel approach that integrates genomic features with structural and functional imaging data to diagnose schizophrenia (SZ). This innovative method utilizes self-attention mechanisms and transformer architectures to capture both neuroanatomical and connectome abnormalities associated with SZ. The research highlights the importance of integrating different data types for improving diagnostic accuracy in psychiatric disorders, particularly SZ, given its diverse cognitive impairments and overlap with other conditions. Structural magnetic resonance imaging (SMRI) provides detailed images of brain anatomy, enabling the assessment of brain morphology and detection of abnormalities. Functional magnetic resonance imaging (fMRI) yields connectome data, capturing brain activity patterns and functional connections. MIGTrans demonstrated an improved SZ classification performance with an accuracy of 86.05%, plus or minus 0.02, offering clear interpretations and identifying significant genomic locations and brain morphological connectivity patterns associated with SZ. This model's ability to attentively integrate genomics with imaging data can significantly enhance our understanding of SZ and improve treatment efficacy. The use of single nucleotide polymorphisms, SNPs, as genomic features adds a crucial layer of information, given their potential in identifying heritable traits related to SZ. The application of transformer architectures in MIGTrans allows for the modeling of long-range dependencies and interactions between genomic and imaging data, providing insights into the complex interplay between genetic and neuroimaging markers of SZ. The findings of this study underscore the potential of multimodal integration in improving diagnostic precision in psychiatric disorders. By leveraging advanced machine learning techniques and diverse data sources, researchers can better understand the underlying mechanisms of complex conditions like SZ and develop more effective diagnostic tools. In the realm of understanding schizophrenia, SZ, a comprehensive approach integrating multiple modalities such as genomics, connectome, and structural magnetic resonance imaging, SMRI, is pivotal. This integration offers a profound insight into the disease by linking macroscopic brain differences with microscopic molecular insights. While previous studies have explored individual modalities or their fusion through simple concatenation, attentional feature fusion, or methods like sparse CCA, graph neural networks, and attentive deep CCA, they may overlook complex intermodality relationships or capture cross-modal interactions implicitly. The Vision Transformer (VIT) has been employed to integrate functional and structural MRI features, providing a nonlinear approach to learning attentive representations. However, integrating imaging with genomic biomarkers poses significant challenges due to their complex nature, differing dimensionality, scale, and intricate relationships. To address these challenges, the authors propose a novel three-way stepwise attentive integration approach that leverages the complementary information across genomics, connectome, and SMRI features to enhance SZ diagnostic accuracy. The integration order, genomics right-pointing arrow connectome right-pointing arrow SMRI, is strategically chosen based on inherent correlations between the modalities. The primary contributions of this research include an attentive integration for multimodal imaging genomics, which integrates genomic and connectome features attentively through cross-modal multi-head attention, prioritizing genomic and connectome interactions, and further integrating with SMRI features, selectively attending to relevant regions, and extracting complementary information from all modalities. Additionally, the study presents a deep learning model, Multi-modal Imaging Genomics Transformer, MIGTrans, which combines structural brain imaging and functional connectome data with genomic biomarkers to enhance clinical interpretability. MIGTrans ranks the top SNPs, identifies significant connections within the connectome, and highlights SZ-specific regions in SMRIs for enhanced clinical insight. The Multi-modal Imaging Genomics Transformer, MIGTrans, is a novel architecture designed to integrate genomic and connectome data with structural MRI for distinguishing between individuals with schizophrenia, SZ, and healthy controls, HCs. The model consists of four key components: genomic and connectome encoders, a structural MRI encoder, and a fusion transformer. The genomic encoder, Phi processes genomic descriptors through a neural network function with two dense layers of 2048 and 1536 units, respectively, activated by the Gaussian error linear unit, GELU. Each layer is equipped with layer normalization and dropout mechanisms to ensure robustness and prevent overfitting. The final layer outputs latent representations of genomic features, encapsulating crucial information for discriminating SZ pathology. Similarly, the connectome encoder, Psi, takes the connectome descriptor as input and learns nonlinear connectome representations through two dense layers with 2048 and 1536 units, activated by GELU, along with layer normalization and dropout mechanisms. The output is a latent representation of connectome features, essential for understanding the relationship between the brain's structural connectivity and genomic data. The structural MRI encoder, Theta, processes the structural MRI descriptor through a convolutional neural network, CNN, consisting of multiple convolutional layers with max pooling operations, followed by two dense layers with 2048 and 1536 units, activated by GELU. The output is a latent representation of structural MRI features, capturing morphological information relevant to the study of SZ. The fusion transformer, Phi, integrates the latent representations from the genomic, connectome, and structural MRI encoders using multi-head attention mechanisms. This module consists of multiple layers, each comprising a self-attention mechanism and a feed-forward network. The output is a comprehensive multimodal representation that encapsulates the combined information from all three data types. By employing specialized encoders for each modality and a fusion transformer for integration, MIGTrans offers a novel approach to understanding the complex interplay between genetics, brain structure, and function in the context of psychiatric disorders. The neural network architecture presented in this research paper integrates two distinct modules to learn functional and morphological representations of brain connectomes and structural MRI images. The functional connectome representation learning module takes a functional connectome C as input and processes it through a dense layer with 1536 units, utilizing the Gaussian error linear unit, GELU, function, layer normalization, and dropout mechanisms. This process yields a latent representation of the connectome, C, capturing intricate brain connectivity patterns, as described by equations 3 and 4. In the morphological representation learning module, a pre-trained 3D dense net 121 is employed to extract features from gray matter density in structural MRI images. The model transforms the input SMRIs through a series of operations, including 3D convolutions, pooling, dense layers, and transition layers, producing a feature vector X that encapsulates the subject's brain morphological characteristics. This vector is then fed into a spatial sequence attention (SSA) module to learn attention across spatial and channel dimensions. The SSA mechanism utilizes a 3D convolutional CONVA 3D layer, a CONVA LSTM layer, and another CONVA 3D layer to capture spatial and channel dependencies within morphological feature maps X. The CONVA LSTM layer, a variant of LSTM with convolutional operations, enables the learning of intricate spatial interactions and relationships across channels. By incorporating tanh and sigmoid activation functions, the CONVA LSTM controls the flow of information and learns from sparse connections between input and state transitions, allowing it to learn both short and long-term dependencies within 3D feature maps. As illustrated in equations 5 and 6, the evolution of the cell state, quart, and hidden state, HT, at each time step t is determined by the input gate, it, forget gate, FT, and output gate, OUT, considering both the current and past feature maps in the input sequence. This mechanism enables the learning of complex dependencies within morphological feature maps. The CONVA LSTM module, governed by equations 5 to 8, integrates convolution and elementwise product operations to update the cell state. The input gate evaluates the importance of input feature maps, while the forget gate determines feature retention from the previous cell state. The cell state is selectively updated by integrating input features and forgetting unwanted ones. The output gate regulates the exposure of the updated cell state as the output. Through learned weight matrices and bias vectors, the CONVA LSTM adapts to the specific characteristics of the data, capturing spatial and channel dependencies within morphological feature maps. The SSA outputs attentive SMRI features, denoted as X. The learned genomic features G and connectome features C are attentively integrated through cross-modal multi-head attention, XMHA, with two heads, referred to as the genomic connectome transfuser. This module emphasizes the fusion of functional connectome from fMRI with non-imaging genomic biomarkers. Genomic features are transformed and passed as query, key, and connectome features are passed as value to XMHA, enabling the model to compute attention between genomic and connectome features through a scaled dot product mechanism, referred to as self-attention. The attention scores are computed as the softmax of the scaled dot product of the transformed genomic features, facilitating selective focus on relevant interactions. The resulting representation is combined with the original connectome features using layer normalization and element-wise addition. The final fused connectome representation, denoted as GC, is obtained through element-wise multiplication with the original connectome features. Equations 9 to 11 illustrate the mathematical operations in scaled dot product attention in XMHA with two heads, as well as the formulation of the genomic connectome attention features. This approach is tailored to extract meaningful insights from the complex interplay between genomic and connectome data, enabling the model to effectively capture intricate dependencies specific to schizophrenia. The XMHA module selectively focuses on relevant interactions between genomic and connectome features, allowing the model to learn a more comprehensive representation of the data. The Genomic Connectome SMRI Transfuser module integrates genomic connectome features with structural MRI, SMRI, features using a cross-modal attention mechanism with two heads, namely the X multi-head attention, XMHA. This module captures complex relationships between imaging and non-imaging biomarkers, specifically focusing on cross-modal interactions. In the XMHA setup, genomic connectome features are treated as query and key, while SMRI attention features, after applying the squeeze excitation mechanism, are used as value. This allows the model to selectively attend to relevant regions in the SMRI features based on the genomic connectome attention features, effectively incorporating complementary information from both modalities. The attention-weighted representations are then fused with the original SMRI features through layer normalization and element-wise addition, ensuring the adaptive combination of information from multiple modalities while preserving the structural characteristics of the SMRI. This process is elucidated in equation 12, which outlines the layer normalization and element-wise addition operations. The fused representations undergo further processing through dense layers, effectively retaining cross-modality features alongside the fused representations and enhancing the model's generalization performance. The feed-forward neural network, FFNN, serves as a classification head, utilizing the fused feature representations to predict target labels. The FFNN consists of two dense layers with 512 and 256 units, respectively, employing the Gaussian error linear unit, GELU, activation function to introduce nonlinearity and capture intricate features. The entire model, including the Genomic Connectome SMRI Transfuser module and the FFNN, is trained end-to-end using the Atom Optimizer to minimize cross-entropy loss. This loss function measures the dissimilarity between true labels and predicted probabilities. The proposed MIGTrans model is evaluated on a subset of the Function Biomedical Informatics Research Network, FBIRN, dataset, containing SMRI, functional MRI, fMRI, and single nucleotide polymorphism, SNP, data from 186 participants. The dataset is preprocessed by selecting a subset of 4,942 SNPs related to schizophrenia, SZ, extracting the lower triangular matrix from the functional network connectivity, FNC, matrix, and downsampling each SMRI to specific dimensions. The model's performance is assessed using 5-fold cross-validation, with various hyperparameter values explored to optimize its performance. The Multi-modal Imaging Genomics Transformer model is evaluated in terms of its performance and qualitative assessment. The model's hyperparameters are set to an initial learning rate of 0.001, dropout rates of 0.5 and 0.3, a batch size of 16, 100 training epochs, L2 weight regularization of 0.005, and L1_L2 bias regularization of 0.005. Quantitative evaluation reveals that the connectome alone achieves the highest performance among single-modal features, with an accuracy of 82.33%. However, combining genomic and connectome attentive features yields a higher accuracy of 82.71%, demonstrating the superiority of multimodal features over single-modal ones. In terms of multimodal feature integration, the proposed trans method significantly outperforms other methods, such as concatenation and AFF, for both two-way and three-way fusion. Specifically, the trans method achieves a 2.11% and 0.99% improvement in accuracy compared to concatenation and AFF, respectively, for two-way fusion. For three-way fusion, the trans method achieves a 1.72%, 1.73%, 1.74%, and 1.75% improvement in accuracy compared to SCCA, AFF, ADCCA, and concat, respectively. This highlights the effectiveness of stepwise attentive integration of imaging with genomic biomarkers through cross-modal multi-head attention. Notably, AFF performs 0.02% lower in three-way fusion compared to concatenation, indicating its limitations in integrating multiple modalities. Qualitative evaluation provides further insight into the model's interpretation and clinical validation. By analyzing attention maps of structural magnetic resonance imaging, SMRI, and important connections in functional connectivity, we gain a deeper understanding of the model's decision-making process and its relevance to clinical findings. The Multi-modal Imaging Genomics Transformer, MIGTrans, integrates genomic, connectome, and structural imaging features through a novel three-way stepwise attentive integration approach. This method leverages complementary information from diverse data modalities to identify significant genomic locations, capture brain connectivity patterns, and highlight spatial regions specific to schizophrenia. By combining these features, MIGTrans surpasses the performance of baseline methods, demonstrating its effectiveness in enhancing clinical decision-making and improving diagnostic accuracy for schizophrenia. The attentive integration mechanisms in MIGTrans pinpoint the top 3 schizophrenia-associated single nucleotide polymorphisms, SNPs, including 7,643,366 rupees, 396,911 rupees, and 13,429,970 rupees, consistent with a genome-wide association study. Furthermore, the generated attention maps for structural magnetic resonance imaging, SMRI, highlight primary regions in the frontal and temporal lobes in schizophrenia samples, contrasting with thalamic regions in healthy control samples. This aligns with clinical observations indicating that individuals with schizophrenia experience significant reductions in thalamic volumes and thickness within the frontal and temporal lobes. MIGTrans also successfully identifies significant interactions in functional brain networks, including the default mode network, DMN, and cognitive control network, CON, which are critical for schizophrenia classification. Notably, the approach reveals denser connectivity patterns in healthy control subjects compared to individuals with schizophrenia, emphasizing the discriminative potential of the attentive integration mechanisms in MIGTrans. Overall, MIGTrans holds promise for advancing our understanding of schizophrenia and facilitating more personalized therapeutic interventions in clinical settings. This review presents a comprehensive overview of research studies and methodologies in the field of multimodal imaging genomics for the classification and prediction of schizophrenia. It emphasizes the importance of integrating different data types, such as structural MRI, functional network connectivity, and genetic information, to better understand the etiology and symptom severity of this complex mental disorder. Several studies have employed advanced machine learning models, including transformers, graph neural networks, and attentional feature fusion techniques, to analyze and integrate these diverse datasets. These approaches aim to identify reproducible markers of brain disorders and predict symptom severity based on multimodal inputs. The Multi-modal Imaging Genomics Transformer, MIGT, model is highlighted for its use of self-attention mechanisms to fuse information from different modalities and predict patient outcomes. This model is compared with other state-of-the-art models, such as the multi-vid model, which uses a vision transformer to integrate structural MRI and functional network connectivity data for schizophrenia prediction. Genetic aspects of schizophrenia are also discussed, with reference to studies that have identified genetic variants associated with increased risk of the disorder. These findings are supported by large-scale genetic studies and databases, such as the GWAS catalog, which provides a comprehensive repository of genetic associations across various diseases. The importance of considering both genetic and environmental factors in understanding the development and progression of schizophrenia is emphasized. A prospective cohort study that investigated the interplay between genetic and perinatal influences in the etiology of the disorder is referenced. Finally, the need for further research in this area is highlighted, particularly in terms of developing more sophisticated models that can effectively integrate and analyze multimodal data to improve diagnostic accuracy and treatment outcomes for patients with schizophrenia. A comprehensive list of 30 machine learning algorithms is presented, categorized into 4 primary groups: regression, classification, clustering, and dimensionality reduction. These algorithms are evaluated using a novel framework that assesses their performance on a diverse set of datasets, including high-dimensional and sparse data. The regression algorithms, including linear regression, ridge regression, and lasso regression, are compared using metrics such as mean squared error, MSE, and coefficient of determination, R-squared. The results show that ridge regression outperforms linear regression in terms of MSE, while lasso regression achieves better performance on sparse datasets. Classification algorithms, including logistic regression, decision trees, and support vector machines, SVM, are evaluated using metrics such as accuracy, precision, and recall. The results demonstrate that SVM achieves superior performance on high-dimensional datasets, while decision trees exhibit better performance on datasets with a large number of features. Clustering algorithms, including k-means and hierarchical clustering, are compared using metrics such as silhouette coefficient and Kolinsky-Haribas index. The results indicate that k-means outperforms hierarchical clustering in terms of silhouette coefficient, while hierarchical clustering achieves better performance on datasets with a large number of clusters. Dimensionality reduction algorithms, including principal component analysis, PCA, and T-distributed stochastic neighbor embedding, TSNE, are evaluated using metrics such as reconstruction error and preservation of local structure. The results show that PCA achieves superior performance on high-dimensional datasets, while TSNE preserves local structure better on datasets with nonlinear relationships. The evaluation framework also considers the computational complexity of each algorithm, with results showing that some algorithms, such as linear regression and k-means, exhibit lower computational complexity compared to others, like SVM and hierarchical clustering. The study highlights the importance of selecting the appropriate algorithm based on the specific characteristics of the dataset and the problem at hand. The findings also underscore the need for further research on developing algorithms that can effectively handle high-dimensional and sparse data. Limitations of the study include the focus on a specific set of algorithms and datasets, and future work should aim to expand the evaluation framework to include additional algorithms and datasets.